Okay, so we're gonna get started now. So this week we're going over cell cycle 2, topic 8. So first up, we're gonna be talking about S phase. Oh, first, can everyone hear me? Okay. No one's saying anything in the chat, so I'll just assume you guys can. Or yeah. So first is the pre-initiation complex or pre-IC. Someone was. I actually can't see the chat. Would you be able?
Yeah, I'll.
Okay.
No, I can get it.
Okay. So after the pre-RC is formed in G1, more proteins are recruited to form the larger complex, which is called the pre-IC. CDK is required for this pre-IC formation. And after the pre-IC forms, DNA polymerase can be recruited. And once this polymerase is recruited, the overall complex is known as the replosome. So then the origin of replication fires and DNA replication can begin, and this is the start of S phase. And also it's important to note that all origins don't fire at the same time. Some happen earlier and some happen later in the in S phase. So then there needs to be suppression of re-replication, and why this is because cells can reform the pre-RCs in the same cell cycle, which leads to reinitiation of replication from the same origin. And this would lead to increased DNA damage and would just cause too much DNA in the cell. And cells don't want to have this extra DNA as it would cause cell death or lead to a cancerous phenotype. And this suppression happens by CDK. Like it said in the previous slide, it is required for formation, but it also is required to suppress, which you can also see in the image to the right here. So CDK phosphorylates ORC, and this provides CDC6 from binding. CDK phosphorylates CDC6 itself, and this gets tagged for degradation through ubiquitin ligases, and it gets degraded in the proteasome. And then CDK phosphorylates various subunits of the MCM complex, and it causes it to get exported from the nucleus. Are there any questions up until this point? Okay. So then I believe she talked about it a little bit in class on Monday about the cohesion rings. So these form around replicating DNA, and these rings keep DNA strands together throughout replication and into M phase. They prevent chromosome segregation during spindle assembly and ensure an equal number of chromosomes for division. So when she was mentioning it in class, they did some experiment trying to see if they loaded in cohesion in G2, if a spindle assembly checkpoint would still be able to function properly, and it was not able to. So this kind of shows that cohesion ring loading must occur in S phase. So then at the end of S phase is the checkpoint that looks for DNA damage and checks if DNA has been replicated completely. And replication can be stopped by this checkpoint temporarily if necessary. And P53 can also be activated here as well, not just for G1. So then before we go into G2, is everyone okay up to here? Okay. So in G2, the cell is again growing and prepping for chromosome segregation, which just means it's inhibiting entry to M phase until the cells have grown enough and have enough nutrients and that there is no DNA damage. And without activation of M phase CDK, the cell will remain in G2 and not enter M phase. So early in G2 phase, the M phase cyclin levels do rise, and they start rising in S phase as well, but MCDK is still inactive at this time. And this is because WEE1 phosphorylates MCDK with an inhibitory phosphate, and although CAK phosphorylates MCDK with an activating kinase, because that inhibitory phosphate is still on the CDK, it's going to remain inactive. And this is important so the cells can continue to grow. So after the cell passes the G2 checkpoint, then MCDKs become active. So CDC5 is an activating phosphatase, and it's partially activated by PLK1 kinase. And when it's partially activated, it can take off the inhibitory phosphate that WEE1 puts on the MCDK complex. And now that MCDK is fully activated, it there's a positive feedback loop that occurs, and it fully activates CDC5, which you could see in the image to the right. So the MCDK is fully activating the CDC5, which causes rapid activation of the M-cyclin CDK complexes. So the transition into M phase happens fairly quickly. And the MCDK, once it's fully active, also inactivates WEE1 to prevent it from keep adding more inhibitory phosphates. Is this clear up to this point? Does anyone have any questions? Okay. So here's just a quick little overview of M phase. So in prophase, that's when MCDK is active. There's a breakdown of the nuclear envelope and the chromosomes condense. In prometaphase, the microtubules grow and shrink towards their respective locations, and it's looking for proper kinetochore attachments. In metaphase, the chromosomes align on the metaphase plate. These chromosomes have proper attachments, and this is where the spindle assembly checkpoint is. And then in anaphase, it's the separation of duplicated chromosomes, and telophase and cytokinesis is where the nuclear envelopes start to reform, chromosomes decondense, and the cell divides. So in the first stage of M phase prophase, these active MCDKs phosphorylate lamins, which are required to break down the nuclear envelope. They phosphorylate condensin, which induces chromosome segregation. So another way to think about this is just the DNA is kind of going from like spaghetti to sausages. And then active MCDKs also phosphorylate various maps and motors that are required for spindle assembly. And then there's also sequential phosphorylation. So phosphorylation needs to occur in a particular order. CDKs, it's important to remember that CDKs are master regulators of the cell cycle, and CDK acts as a priming kinase for downstream kinases. So one example of this is how CDK acts as a primary kinase for the substrates of PLK1 kinase. So the substrates of PLK1 need to be phosphorylated by CDK, which you could see at the top part of this figure. So then it could bind to the PLK1 kinase here. So then PLK1 can bind to its substrate. And PLK1 plays an important role in the G2 M phase transition, which if we go back a few slides, it was here, the partially activated PLK1 is what activates CDC25. And then it's also important in the spindle assembly checkpoint. Well, just spindle assembly in general. Any questions before we go into the spindle assembly?
Okay, so now we're on the spindle assembly in M phase.
So spindle assembly begins at the beginning of M phase, during prophase, specifically. And during spindle assembly, there are three types of microtubules that form from the spindle. Those three are aster microtubules, interpolar microtubules, and kinetic core microtubules. So for aster microtubules, they attach to the inner side of the cell membrane, seen in the green on the picture on the right. Interpolar microtubules overlap in the middle of the spindle. The motors hold these microtubules together, seen in the pink and the little black dots in the middle of this picture. And the kinetic core microtubules extend from the spindle poles and connect at the kinetic core of the chromosome, shown in the blue, which connects to this little red part of the chromosome. So now going into what kinetic cores are. Kinetic cores are the large group of protein, or are a large protein structure on the chromosome, and they're built on the centromere region of the chromosome. The kinetic core microtubules that extend from the spindle poles attach here. So this can be seen a little bit more clearly in the picture on the right. The green microtubule is attaching to this red protein on the chromosome. Does anyone have any questions about what we just went over? Okay. So building the spindle with the help of microtubule motors. So this is done through a theory called push and pull mechanisms. Minus end directed motors such as dynein are anchored to the cell membrane, shown at this little part of the picture right here. These will walk towards the minus end of the microtubule, which is towards the spindle pole. Oops, sorry. And these will be responsible for pulling the spindle poles apart. So as dynein walks towards this minus end, this spindle pole is going to come closer and closer to the cell membrane. On the other hand, plus end directed motors such as EG5 sits on the overlapping interpolar microtubules seen in this kinesin 5 right here. They walk towards the plus end of each microtubule, and this causes the spindle poles to be pushed away from the center of the cell. So now the spindle assembly checkpoint. The purpose of this checkpoint is to check for proper spindle attachment at the kinetic cores from opposite spindle poles. It's essentially checking for tension on each sister chromatid, and once tension is felt on all of them, anaphase will begin. So now the mechanism of the spindle assembly checkpoint. So to start, before this entire process that you see on the right, there are two proteins called MAD and BUB, and these will be responsible for sensing tension on all sister chromatids. So after they sense correct tension, CDC20 will be released, and it will go and bind to APC, which is this blue protein on the left-hand side of this picture. So APC is a ubiquitin ligase, and essentially that means it tags proteins with ubiquitin to be sent to the proteasome to be degraded. So after CDC binds to APC, this complex, it is now active and it's able to ubiquitinate securin, which is this brown orange protein on the picture. And securin's job is to inhibit separase activation. So once securin is degraded, separase is now going to be active. Once separase is active, it will go ahead and cleave any cohesin, and this is going to release the sister chromatids from being stuck together. Now that the cohesin is gone, chromosomes are now going to be able to be pulled to the opposite sides of the cell, and this marks the end of M phase into anaphase. And a key point here is APC degrades M cyclin. The cell cannot finish dividing with M cyclin CDK activity. Any questions on the mechanism that anyone has? No? Okay. So now going into anaphase. There's two parts of anaphase A and B. During anaphase A, this is when kinetochore microtubules are shortened and the minus N motors are going to pull the microtubule away from the center of the cell. Wait, yeah, sorry. So part of the microtubule that is embedded inside of the kinetochore is going to be degraded at the plus end. And this is how the microtubule is shortened and allows the chromosome to be pulled towards the pole. So anaphase A essentially just pulls the chromosomes towards the poles. Now going into anaphase B, the poles are pushed and pulled apart towards the outer edge of the cell. So going back to dynein motors, which was a minus end directed motor, those are going to be anchored on the cell membrane, and these are going to be pulling the spindle poles towards the edge of the cell membrane. And then EG5 is still going to be in these interpolar microtubules in this overlapping region, pushing the spindle poles away. And in order for this to occur, the interpolar microtubules are going to have to lengthen themselves at each plus end in order for EG5 to correctly walk in the plus end direction, allowing the poles to be pushed away. So now going into cytokinesis, a contractile ring made of actin is going to form between the new cells. This pinches the two cells together, creating now two cells. And during this, APC will be targeting M-cyclin, and this will cause any M-cyclin CDK activity to drop at the beginning of anaphase. This is important because this drop in activity causes the nuclear envelope to reform, which is important when the cells are becoming cells again and are not dividing. It causes spindles to unassemble and the chromosomes to decondense. And that was all for today. Does anyone have any questions on anything we went over today?
If not, we're just gonna end the recording and then we'll take any problem set questions.